Professor Ponchai Matangkasombut, Professor Yongyut Yuttawong, Dr. John Randell, Professor Alex Shalek, and the Dean of Faculty of Science, Mahidol University, and distinguished guests. Welcome to the 2024 Human Cell Atlas Single Cell Omics in-person computational and experimental design workshop. So before we begin, I'd like to quickly notify you that all workshop participants may be photographed or video recorded during this event, and by entering the event premises, you consent to media recording and dissemination on various websites and social media platforms. This workshop was organized through a huge collaborative effort by the Human Cell Atlas, Mahidol University, the Massachusetts Institute of Technology, or MIT, the Broad Institute, and the Waradon Institute. We would like to thank the workshop staff and training leaders, as well as Mahidol leaders and staff for all the time and effort that they put into organizing this workshop. We'd also like to thank our very generous supporters, including the Chan Zuckerberg Initiative, who is our major supporter. We'd like to thank 10x Genomics and Bioactive, as well as Parse Biosciences and RI Technologies. A huge shout out to both 10x and Parse Biosciences, who very kindly sent us free kits to use for our experimental group. And we also like to thank BD Biosciences, MGI Tech, Genomics, Amazon Web Services, and Biodesign for your generous support as well. So this is today's schedule. The opening session is scheduled from 9 to 10:40. And then we're going to have a quick group photo here at 10:40 to 10:45. So please don't run away yet. And then we have a coffee break at 10:45 to 11:10. At 11:10 sharp, we'd like all pre-registered workshop participants to reconvene here in this room. Please note that the workshop from 11:10 onwards, it's only reserved for pre-registered participants, so it's not open to the public. All right, so without further ado, please welcome the Dean of the Faculty of Science, Mahidol University, Associate Professor Prasit Suwannalert, for his opening speech.
Morning, Professor Ponchai, Professor Yongyut, and esteemed guest, distinguished speaker, Professor Alex Shalek, Professor John Randell, Professor Ponpan Matangkasombut Chupong, Professor Varodom Charoensawan, Dr. Waradon Sungnak, and all participants. On behalf of the Faculty of Science, Mahidol University, I'm honored to welcome you to the workshop and training in the theme Single Cell Omics Techniques. This training initiated by the MU Single Cell Research collaborates with the partner from MIT, the Broad Institute, the Waradon Institute, and the Human Cell Atlas. At the single cell biology is a very interesting and is quite important in the many diseases. I give example in the cancer who got cancer, they act with the same of the cancer cell type, but not easy to treatment because in the different of the cell and the function, the knowledge of the single cell apply to the disease and help to the screening and the treatment. The treatment is not only treat the patient, but also treat her and his family. That align with the global impact and well-being. In the finally, I am express my sincere thanks the organizer, sponsor, and participant, and hope that this conference is not fulfilled in your the practice, but also help you initiate and expand the collaboration in the future. Thank you.
Thank you so much. Up next, it's my pleasure to introduce Dr. John Randell. He's the Chief Alliance Officer of the Human Cell Atlas Initiative, and he will be giving us an overview of the Human Cell Atlas. Thank you.
Hello, everyone, and welcome. My name is John Randell. As you heard, I lead the Executive Office, which is a small but very busy executive staff that supports a very large international consortium known as the Human Cell Atlas. And you'll hear much more about HCA science from the subsequent speakers, but my goal here is to really introduce you to this project and to sort of give you an update on where we are in sort of the arc of our history, as you were. So I'll begin with the mission of HCA. So I'll begin with the mission of HCA, which is this: to create comprehensive reference maps of the types and properties of all human cells as a basis for understanding, diagnosing, monitoring, and treating health and disease. And so the key link here is that knowing our cells, knowing the cells in the human body is really critical for understanding disease. And that's because we really want to understand, if we want to understand how genetic variants act in disease, we need to know the cell types that those variants are active in and what the functions of those cells are in order to understand the link to disease itself. Now, you would be forgiven for thinking that we actually know everything there is to know about the cells in our body. After all, there's been many, many decades of study in cell biology. But the problem is we actually don't really have a complete understanding of all of the cell types that make up the human body. In some sense, this is understandable. There are about 37 trillion cells in the body. But if you read a standard biology textbook, you might see something like a few hundred cell types that make up all of the organs and tissues in our body. But as more and more information emerges, particularly over the last decade or so, we realize that this is a vast underestimate. And in fact, even in the brain, recently there have been over 3,000 subtypes of cells identified. And so understanding that sort of complexity is really the goal of this international project. And so how are we doing this? Well, historically, cells have really been characterized by a number of attributes. Here you see by their structure, their location, the tissue that they're located in, their function, the molecules that they secrete, and so on. And these are, of course, all really important attributes, but just in the last dozen years or so, scientists have developed very, very powerful new techniques to study cell types at the level of the individual cell. And this is uncovering a lot more heterogeneity and a lot more variety than was previously appreciated. And you'll hear much more about these techniques as we go along, but the foundational technique here is single-cell genomics, and that's really what this entire workshop is about this week. So in single-cell genomics, rather than needing to study an entire tissue sample at once, which might be tens of thousands of cells, so taking an average across the entire tissue sample, we can actually dissociate the sample into individual cells, and then techniques have been developed to quantitatively measure the level of gene expression at the level of an individual cell. And you can do this for every single cell in the sample at once. So you get an enormous amount of data here, and you can study the gene activation, i.e. the RNA expression in that cell across the 20,000 plus genes in the human genome. And so really what you get is this sort of many thousand dimensional space of variable gene expression across the genome in every individual cell, and that is called the transcriptome, and that is sort of a hallmark of a cell type. So by understanding what the transcriptome looks like in an individual cell, you can then identify what group of cells or what cell type that cell belongs to. And again, you'll hear much more about this in the subsequent talks. But you can imagine that with 37 trillion cells in the body, doing this at scale is an effort that's way beyond the capabilities of a single research group or even a group of research laboratories. And so this really needs to be a global initiative. Our membership in HCA, which was founded in 2016, continues to grow. So today we have nearly 3,500 members at 1,800 institutes in over 100 countries now. So very much a global international effort. This map shows you how those members are distributed. I will say that they are all supported by regional networks, and particularly the HCA Asia network, which is actually our earliest regional network, has been very active since the very founding of of HCA. It's had an annual meeting. I've attended the last couple. They're really quite exciting events. The next one will be in Hong Kong in December. I believe Ponpan will tell you more about that meeting. And HCA Asia has really given us a model for how to foster cross-regional collaboration in single-cell genomics, and we've sort of replicated that in Latin America and more recently in Africa and the Middle East. And so these regional networks are really powerful venues for collaboration and for really doing sort of collective science. In addition to the regional networks, though, I'll give you a quick overview of how we are organized. There is a 36-person organizing committee. This is the scientific steering group for HCA. It's very much a representative international body. They meet regularly to set policy and to review the scientific outputs of HCA. They've established several working groups: data analysis, ethics, equity, standards and technology, and the newest group overseeing our data ecosystem, the data platforms, which I'll tell you a little bit about in a second. And I just want to acknowledge, since we have Dr. Alex Shalek here, he's a co-chair of the Equity Working Group and has really done a lot to advance equity in both sort of participation and in data within HCA. The real heart of the research and what I'll call atlas building in a second is the 18 biological networks. These are research networks that HCA has created to study individual organs and tissues. So we're sort of taking a divide and conquer approach to generating a map of the entire body. So each of these biological networks focuses on one particular organ or system or tissue. They number anywhere from 50 to 650 scientists, so these are very large networks, and they're responsible for data curation and for the building out of these objects that we call single-cell atlases. And then finally, as I've already mentioned, there's a small executive office, which I lead, and it is based at Singapore, at Japan, in the United States, and the United Kingdom, and we provide sort of the organizational support for the entire global project. I want to return to the 18 biological networks. They're represented by the icons here. You can see that they range from adipose, heart, nervous system, skin, musculoskeletal, gut, and pretty much covered most of the tissues in the human body. This, again, is really the heart of our approach to building a human atlas, a map of the entire body, identifying all of the known cell types in the human body and many that are unknown. The charge to each one of these biological networks is to create atlases covering their tissue of interest. And so our approach to building the entire body is to sort of stitch together the atlases that are produced by these 18 biological networks to create a whole body map. And I want to pause for a second and explain what I mean when I say atlas. So essentially, and when we talk about an atlas, what we're talking about is asking each of these networks to curate data sets, anywhere from 6 to 60 or more data sets generated around the world in very much a distributed grassroots manner. They need to be aligned in terms of the file formats, the metadata standards, the quality control standards, and so forth. Then once you do that alignment, you can actually combine all those data sets into sort of a unified data object that then becomes a sort of an atlas for that particular tissue. And the idea here is that that gives you the statistical power to really do very rigorous science, to uncover cell types that may be rare or previously undiscovered, or to tease out the differences between closely related cell types. And by doing that, you can get a very high resolution picture of a particular tissue. And that's all I'm really talking about when I'm talking about cell atlases. 2 of these biological networks have already published their first atlases, and I want to say the first version of their atlases, because this is a very iterative process, as you might imagine. One of those, the first was the lung, which published their first atlas in 2023, actually almost exactly a year ago. And then they were followed last October by the nervous system, which produced 2 brain atlases. And I'll point out that these were actually the product of the NIH Brain Initiative, particularly the Brain Initiative Cell Census Network, which has now become the Cell Atlas Network. So we've actually been partnering with the Brain Initiative to include these brain atlases as part of the HCA corpus. And so this is just one example of how we can partner with other consortia and other initiatives in our quest to create a whole body resource. So those were the first 2 to be published, but many more are to follow. I think the eye, organoid, kidney, gut, immune, and breast atlases are nearing completion, and we expect them to be pretty much complete again with the first draft this year, and then the rest will follow with their first initial drafts in 2025. But I keep on calling these the initial draft atlases, and there's a reason for that. which I'll get to in a second. But first, I want to say that you can find both the data sets that are making up the atlas as well as the atlases themselves at data.humancellatlas.org. This is our data portal. There's an Explore Data button here, which you can use to search for individual data sets. You can see that there are about 455 data sets currently in the data portal. But then as atlases are released by the biological networks, they will appear and be accessible through the biological network icons down here in the data portal page. And these blue dots signify that the lung and nervous system networks have already released atlases, as I just described to you. But as I mentioned, we're really only at this first draft stage here, where we imagine that the first drafts of all of these atlases combined will constitute roughly 100 million cells. But we expect that there will be a lot of gaps. Those will be both anatomical gaps, tissues that don't have full representation in these initial drafts, as well as gaps in human diversity, both genetic diversity, gender, ancestry, and so forth, but also diversity in geography, age, and other dimensions of human variation. So the ultimate goal, which will take many more years, is to really get to a comprehensive atlas, which could contain billions of cells, but really is fully representative of not just human anatomy, but also human diversity as well. Again, genetic diversity, but also environmental diversity and other forms of human diversity. So in addition to those elements, and I said I've been focusing a lot on single-cell transcriptomics so far, but there are other types of data that will be worked into the atlas over time. Other modalities, multi-omics, including, for example, chromatin structure, perhaps protein, other forms of omics. There are techniques that actually enable you to gather this information in the spatial context, so you can actually start to build a three-dimensional map of cell types. And then finally, I've already mentioned the element of human diversity, which is really paramount, and from day one in HCA has been really important, and is a big part of the reason why we've put so much support into the regional networks, including HCA Asia, as well as training workshops such as this one, to really make sure that we have the capacity around the world to do this research. But even in the first drafts, HCA is already leading to medical advances, and I'm not going to go into a lot of detail here. But some of my favorite examples include cystic fibrosis, where I mentioned rare cell types earlier. There were a pair of studies in 2019 that identified a rare cell type called an ionocyte that actually turns out to be the main mediator of cystic fibrosis and was previously completely unknown to science. And so this is an example of the power of single-cell genomics to uncover cell types that were just previously not even discovered. There are also examples for melanoma, and the HCA community did a lot of work in the early months of the COVID-19 pandemic to really analyze across the entire human body cell types that were likely to be infectable by the COVID-19, by SARS-CoV-2. You'll hear more about other meetings. We do a lot of meetings around the world every year, including regional network meetings. You'll hear about the next HCA Asia meeting in December from Ponpan. The next major meeting coming up is actually our annual general meeting, which will be held this year in Milan, Italy. You're all invited. You're also, by the way, all invited to join HCA. The only requirement is that you support our principles and our mission, so anybody can join HCA. We would certainly encourage you. We would love to see you in Milan as well as in Hong Kong towards the end of the year. I do want to note that if you want to register, there are early bird prices that end at the end of this month, so I would urge you to do so soon. So before we get to the sort of more science-heavy talks, I just want to sort of review the goals for the workshop that's happening this week. It's really a pleasure to be here. It's a pleasure to be partnering again with Mahidol University, as we did 2 years ago for the HCA Asia meeting, also here in Bangkok. There are 2 tracks. There's an experimental track and a computational track, although we try to foster crosstalk whenever we can. The goals of the experimental track are to introduce you to single-cell sequencing techniques, and particularly how to apply those to specific biological questions, how to actually perform the preparation of samples for single-cell sequencing, including a number of steps you see here, and then understand sort of quality control and the techniques for cleaning up the data. On the computational side, obviously introducing you again to the world of single-cell transcriptomics, how to perform data analysis with single-cell data, and how to draw sort of rigorous biological conclusions from the data, going from the raw data to what we call gene expression matrices and beyond, and then understanding sort of why you choose particular algorithms and how you compare algorithms, and then, of course, also some hands-on lab exercises. But the overall goal of this workshop is really to create a fun environment. What we've really found in past workshops elsewhere is that we see a lot of sort of collaboration starting to occur over the course of the week, which is a lot of fun to see. So we really want to build a community both here in Asia, but also connect you all to the larger HCA community and encourage you to find partnerships going forward to advance your research. And so building that community capacity across Asia, but also across the world is one of the primary goals. So with that, I will end. I think if there's any questions, I can take them. Otherwise, we can go to the next speaker. Thank you.
All right, thank you, John. Next, I'd like to introduce our next speaker, Associate Professor Ponpan Matangkasombut from Mahidol University. She specializes in single-cell omics in human immunology research, and she also serves as an HCA Asia steering committee member, and she will be giving us an overview of HCA Asia.
Thanks so much, Wim. And I'd like to welcome all of you again to our very own home campus at Faculty of Science, Mahidol University. So very nice to see all of you here. So today my job is to talk about Human Cell Atlas Asia. I work here at Mahidol, and my colleagues, Varodom and Varodom, will later talk more scientific talk on our work. So talking about Asia, Asia is a home to 60% of world's population, about 4 billion people, and the world's most populous country in India and China, and has abundant genetics, ethnic and cultural, environmental and socio-economic diversity, as John previously mentioned. However, the genomic data of Asian population are very underrepresented. So as you can see here, only 1 to 2% of genomics data in the world is of Asian ancestry, curtailing the clinical utility of genomics data in Asian population. So with the knowledge that John previously mentioned, that HCA, or Human Cell Atlas, is very fundamentally help our understanding of health and disease, having the Human Cell Atlas will help in diagnosis, signatures, new biomarkers, regenerative biology, targeted treatment, disease mechanism understanding, for example. However, having only genetics data and HCA data from such a small group of Asian population is not very applicable for the whole global population. Therefore, it is very important to take into account the diversity, the ancestry diversity, geographic environment, and also in social context. With the value of HCA, we value community and diversity. Within the white paper, we also stress that HCA is committed to genetic and geographic diversity and equity at every phase. If you look at the membership of this HCA map, you can see that we have members all over the world. But if you look closely, most members are concentrated in America and Europe, more in China, but not so much at other parts of the world. We're underrepresented. So if we zoom into the Asia, we can see the distribution here on the map. So therefore, it's very important to have this diversity. HCA Asia was established in 2018 to 2019 as the first regional network. We now have over 400 members from 19 countries, from almost 300 institutes. That's followed by regional network in Latin America and Africa. So working as a HCA steering committee, we value community building, strengthening regional research alliance, showcasing regional science, identifying synergy and promote collaboration, and we also try to diversify the HCA We have one representative from each of the participating countries, from Thailand, India, Japan, Korea, Singapore, Bangladesh, and also China. So what's the job of HCA Asia Steering Committee? The HCA Asia Steering Committee liaises with global HCA and coordinate annual meetings and workshops such as this one. We collaborate with equity working group, ethic group, and communicate with public as well. So community building is through flagship projects. We have flagship projects on healthy donors, which we'll talk about in a little bit, on cancer, diabetes, infectious disease, gut biology, and genetic diversity. With this, we can co-apply or find support from funders, from local funders, philanthropists, and foundations, and could be international funders. We coordinate data submission to HCA data portal in order to achieve equity and diversity with clinical benefit that would meet local society needs. So we organize several HCA Asia meetings every year. This year is the 8th one, and as John mentioned, we organized the 6th HCA Asia meeting right here in Bangkok in 2022. Last year we have in Calcutta, India, and this year, mark save your date, we'll have it in Hong Kong, so on December 3rd and 4th, and we plan to open registration soon, probably within this week. So with these annual meetings, we discussed among our community, and we found lots of strength in the HCA Asia community. So our strength include the active single-cell research are ongoing in several countries. We have shared interests in the field of local importance. We have genetics and environmental diversity. We have access to very unique disease cohorts. We have similar time zone, this is also important. However, we also have faced many challenges, such as the cost is quite high for doing single cell, and more than half of our nation's members are considered low and middle income country. The distribution of experimental and computational specialists need to be improved. We also have lack of central governing and funding body, which includes also funding, central funding government in tissue and biobanks. We have varying local regulations such as in ethics and in data release policies. And we are shifting towards translational to disease quite early and have less support in basic science on healthy sample, which are very crucial for comparison. At any rate, we have lots of opportunities. So we are organizing the annual meetings and trainings and workshops in order to help disseminate this knowledge and technology transfer. We need more data harmonization and standardization. We need coordination through common interest projects. We promote equity, gender balance, and ethics. We can co-apply for support of local and international funding. And there's more and more technology advancement that drives the cost per cells down. So I'll give you an example of a very important first HCA Asia flagship project called Asian Immune Diversity Atlas, or AIDA. So AIDA started from Xiam, Woong Yang, and Jay from Singapore, Korea, and Japan. And then we also in Thailand, Varodom, myself, and India, Partha also joined later in the AIDA phase 1, which was funded by CCI Seed Network. We're also expanding to collaborate more with Yulia from Russia, Abdullah from Saudi Arabia, and also Sri Lanka. So the first phase we just finished, and manuscript is under review right now. In the first phase of AIDA, with our 5 countries, we have landscape of immune variation across healthy Asian donors from India, Japan, Singapore, South Korea, and Thailand. With this, we profiled over 1.5 million single cells from over 600 Asian donors, one of the biggest single cell data set across populations. So one of the most diverse single cell data set, enabling research on the impact of subcontinental diversity within Asia on immune phenotypes and disease comparison. So with this, I just show you very briefly that we found variation in healthy baseline across Asian population and validated with orthogonal assays cohort. For example, monocytes cluster, we found less monocytes in the Thai population compared to others. This bar represent each country and also each ethnicity, self-report ethnic within country. Singapore, so Singapore Chinese, Singapore Indian, Malay, and this is Korean, and this is Thai. With this, we also find substantial impact of sex, genetic ancestry, self-reported ethnicity on the immune cell proportion. So with that, I'll conclude this talk with asking you to join the HCA Human Cell Atlas. You can scan this QR code or go to this website, and the membership is free and open for everyone age more than 16. With the HCA, you also get commercial discount from partnership program. If you actively contribute to the HCA research projects, and you can also inquire this discount directly by reaching the companies. Most of them are outside at the booth. So again, save the date for HCA annual meeting, December 2 to 4 at Hong Kong. Registration is opening soon, so save the date first. Thanks very much.
Thank you so much. So up next, our next speaker is Professor Alex Shalek. He is a biomedical engineer from MIT, and his lab's research is focused on elucidating the cellular and molecular features that inform tissue-level function and dysfunction across human health and disease. And he will be talking about the overview and impact of single-cell and spatial genomics. So please welcome Alex.
Thank you all so much for joining us, and thank you for having me here. And I'm sorry I couldn't make it to the last HCA meeting. So John gave the overview and talked a lot about, what the HCA is doing. I'm here as somebody who's part of the HCA, as somebody who's deeply interested in creating partnerships with other scientists and learning about what you're doing and figuring out how we might work on it together. Because there's so much that's going on in the HCA, what I thought I'd do is, based upon the prompting that I got, is give you a little bit of a sense of my journey, how I got here, what I'm interested in. And there's lots of science that we do. I'm looking forward to talking to you all about it and finding places where we might be able to work together. So at a broad level, what our lab is interested in is the idea of balance in our tissues. We all know that balance is incredibly important. If you have too much immune activity, for example, you can get an autoimmunity or you could get tissue damage. And if you have too little, you could become susceptible to opportunistic infections or the outgrowth of cancers. So what we do within the lab is we try and figure out what defines homeostasis. What are the deviations that are induced by environmental exposures or infections or things of that nature? Then we try and figure out how we can rebalance the system so that we can cure people or buffer it in the first place prophylactically. And to do this, we spend a lot of our time developing tools that help us understand what's happening within our bodies at a cellular and molecular level. So, like many of you, we're very interested in the diversity of different cell types that exist in our bodies. What different types exist, what qualities they possess. When we're thinking about immune cells, we care a lot about the diversity of what they might target. We care about things like durability and location. Now, the funny thing is, if you think about how I actually got into this originally, I wasn't trained in biology. For Truth in Advertising, the last time I took a biology class was in high school. So the reason I started doing this is because at the time I was speaking to people at the Broad, like Aviv Regev, who said, well, Alex, you can do genomics, and the reason you can do it is you don't need to know what you want to focus on. You can simply measure everything. So if you take a set of cells and you throw them into a tube with some perturbation, imagine this is a pathogen, anything that you want, and you take a control, you can now go look at the expression of all 20,000 plus genes, so you don't have to make assumptions on what's most important. And through looking at differences between the cells that are exposed versus the ones that are not, you can go do integrative analyses that will help you to figure out what's causing these cells to respond. So the idea is we use some correlation, and then we go do some validations, and this tells us how biology is responding to that particular infection. But in doing this, what I realized very quickly was that we were making a very strong assumption. And that's that every cell in the tube is identical to one another, in the same way that I could assume that everybody in this audience is identical to one another, but you're obviously not. Now, the reason this was a problem for me is that, as you know, when we do genomics, we do correlations, but then you have to go prove that those are right. And every time I tried to do this in the lab, it actually turned out that this didn't work. So the first experiment that we tried to do was taking dendritic cells that were exposed to a bacterial Armageddon, so much bacteria that you couldn't imagine they were going to respond differently. And yet we'd see cells had very different levels of RNAs we thought were important. So some cells have thousands of transcripts shown here as purple points. Some had 0. If we looked at proteins, sometimes there was a lot of it in the nucleus. Sometimes there was very little. You know, it wasn't just this system. When we went and looked at cancer cells, sometimes we would kill 90% of the cells in a patient. Sometimes we'd kill 50%, but we never got 100. And even if we took what the biologist told me were the most pure population of cells we could imagine, so some naive T cells, and we put them in a bath of cytokines, what we saw was that we could only make 40% of the cells that we wanted. Now, to my biology colleagues, this was awesome, because 40% Th17 cells, so you can go do all the assays that you want. To me, it was a huge problem. And why is it such a big problem? Well, if you think about what we're fundamentally doing here, we're taking one system, we're applying one perturbation, and we're getting lots of different responses. And so there's no way to imagine you can make a model that works if one input gives you tons of different outcomes. So in trying to understand what drove this variability, we sat down and we started to think about it. The first thing that came to mind was maybe it was all technical. Maybe we were inefficient in our lysis, maybe we didn't amplify things properly, and so we got things where our measurements just didn't match because we didn't do a very good job. Now, this isn't the hypothesis we wanted, but as I said, I wasn't trained in biology and I was doing biology experiments, so it's entirely possible that all of this was my fault. The second thing that I like to think about, because I came from chemistry and physics, was this idea that maybe it's just stochastic or quantum. Maybe it's that at a particular moment when we lyse the cell, one cell wasn't thinking about something, another cell had just bursted and created transcripts, and so there was a difference. In the same way that many of you are probably thinking about what I'm talking about right now, but some of you are wondering what's for lunch and when the coffee break is, but a moment later we'd all be in the same conversation. But the last and most tantalizing hypothesis to us was that maybe this variability was actually real. Maybe there was something interesting here. Maybe the reason the cells didn't look the same was because they weren't actually identical to one another, and there were interesting, meaningful differences. And so what we began to wonder was, instead of treating all these cells as though they were identical because the biologist told us they were identical, could we instead take each one, profile it independently, use this information to find patterns in the data that would then help us to better understand how cells respond? And so I think like an easy way of thinking about this, if you don't think about this very often, is to think about getting information from each and every one of you. And so I apologize, I have the US up here because I have the slide ready, but imagine that we were looking at demographics. So we were profiling information about lots of different people. And we looked at things like, what are the percentage of people that have advanced degrees, what's median income, what's their commute time, what is household rental. Well, some of these things are going to be associated when we go look. So for example, advanced degrees and median income. And what we'll see is that by degree, median income usually goes up. And so we're going to try and do the same thing through genomics. We're going to try and look for patterns across cells and RNAs, and we're going to use that as a way of discovering what are the inner workings of them and how are they doing the biology that they need to do. So I have a couple technical slides, but I want to show you these because I think it's really important, because this is how we worked through it originally. So this is some of the first data we ever generated. These are 18 bone marrow-derived dendritic cells stimulated with LPS. What I'm showing you here, in case you're not used to looking at these IGV traces, are single cells. We have 18 rows here. Each one is a cell, 2 empty wells, and 2 population controls. At the bottom, I'm showing you a series of genes. Each one of these genes has fat bars, which are the exons, which are the coding part of the proteins, and introns, which usually get discarded, but not always. What I want you to see from looking at this data, because we don't often look at data anymore, we just get a matrix at the end and then we start analyzing it, is what the data really looks like. And so when you look across our 18 cells right here, what you can see is that there's a lot of variability. Sometimes we get lots of reads, sometimes we don't. Sometimes things look a little bit more consistent. But if we look at our populations, things look relatively consistent, which is a good first sign. And if we don't add cells to our tubes, we don't get reads to the genome, which is a really good control. So instead of looking at this data right here, if we want to try and get a sense of what we're actually measuring, we can get a little bit more quantitative. And the way we do that is we look at how many reads we're getting over a gene divided by the length of that gene per million. And what this does is it lets us estimate gene expression. So now what I'm showing you is each point is a gene, and I'm showing you for one population of 10,000 cells, what do we estimate expression to be versus the other. And what I want you to see when we look at 10,000 cells is that we get very beautiful correlations. So I take one group and I look at what's average and take a different group and see what's average, we get the same thing. But most importantly, what this is telling you is that we can do very reproducible experimental biology. So maybe it's not all technical. But when we look at our individual cells, what we see is they're tremendously different from one another. So we see in one cell genes that are highly expressed. This is a log scale, so thousands of copies in one cell, but none in the other, and vice versa, but some things that look similar. And as an initial sanity check, if we just average those 18, we start to get back what we would see in the population. Now you might ask, well, what are the things that are shared among these cells, and what are the things that are different? When we go look, it starts to make sense. The things that were on the diagonal that all the cells share are structural proteins and ribosomes, things that we think are really important for the cell to survive. If we look off diagonal, what we see is the things that are most highly variable are immune response. And remember, I told you this is a dendritic cell responding to bacteria. So what's so funny is this is normally what we throw away, but we're seeing that there's a lot of potential information here. Now, before we get too excited about these differences, we have to convince ourselves that what we're looking at is real biology as opposed to technical. And so later today, when we talk about how we're going to work through the workshops, we'll get into the nitty-gritty of this, and we'll do it over the next couple days. But I'll just suffice it to say that the way in which we thought about checking this was to go use orthogonal approaches. So here I'm showing you RNA fish, an in situ method where we go and hybridize directly to RNAs. And what I want you to see is things like beta-2 microglobulin, part of your immune response, is showing up in all of our cells just like we thought by sequencing. But some of these immune genes like CXCL1 that were 1,000-fold higher in one cell versus another are showing exactly the same patterns when we look at RNA. Now, what this has told us so far is that it's not totally technical, that there is some variability. But what we don't know yet is it stochastic, is it random, or is there some structure in it? And the reason that we're all here today is what we found is actually that there is some structure in this data. What was driving all the variability that we saw is that 3 of our cells were different than the other 15. It turns out they were at a different maturity state. Once we found these cells were different, we could go find markers, we could sort them out, we could validate them. And from looking at this more subtle variation, it turned out we were able to find cell circuits. And that's because it was driven by these correlated genes right here that were either high in a cell or low in a cell. And so what that meant is they all went together. They were all showing up at the same time, so they probably had a common upstream driver. And so we went and validated that and proved that the set of correlated antiviral genes was driven by some transcription factors and that we could figure out the circuitry. So the reason I think this is so cool is it sets a framework for the things that we're thinking about today. We can discover cell types, markers, cell circuitry, and other interesting things that we might want as we apply it to human health and disease. So I showed you there are 2 cell types and different programs going on, even in 18 identical cells. So as John started to talk about, there was this idea that came out of it immediately, which was, could you try and figure out this atlas of the body? As somebody who was trained in physics and in chemistry, the idea of a periodic table really appeals to me. And so if you think about it, we have this reference of, and this is not on, there we go, of 30 trillion cells. My textbooks are the ones that said there were 300 because I actually had to go back and look at them. It's nice that my mom saved them from high school so I could find this. But as John said, we're finding lots and lots of different cell types in the retina alone. If you're thinking about what we would want, to help people, to really think about, the fundamental reference, we'd want to know what types of cells exist in your body. We'd want to know what states they can assume. We'd want to understand what locations they exist in, what the transitions are that are possible between them, how they relate to one another in terms of development, and finally, how they change in disease, because this is really important for us to think about how to counteract that or how to fix it. And in many places, we're doing things where we have information that's decades or even centuries old, which prevents us from doing the kinds of medicine I think we'd like to do. So as John also brought up, it's really important to also highlight that cells are the machines that act on your DNA. So if you don't understand how the machine is using the instructions, it's really hard to understand how you get to a disease. So I, like many of you, are interested in the idea of having this reference, creating this human cell atlas. One thing that I think John brought up, but I want to emphasize is, I am not the HCA. John, I guess, is the HCA, but that's a discussion we can have later. But everybody here is the HCA. It's a grassroots movement of scientists that are interested in creating this. So I have no more ownership over it than any of you, and delighted to have you all be part of it, and delighted to create networks so we can all work together to try and figure out what's going on here as a way to understand, diagnose, monitor, and treat disease. So, as was brought up before, I'm deeply involved in the Equity Working Group, which is really focused on promoting equity. I do a lot of global science research, as you'll find out in a second. It's really important to me that we don't do helicopter science, but that we show up, we create capacity, and we enable people to do work. We really think about inclusive representation, participation, and equal benefit. There was, a great talk that was just brought up talking about the diversity in the human cell atlas in the immune system with respect to Asia. I really do believe that we need to profile diversity to get a fundamental reference, because otherwise it's not a reference for all. But my particular passion is really in focusing on participation and making sure that the data benefit people, because I think in lots of places it's easy to say we want to get samples, but an important part is to actually partner with people to understand what kind of science they want to do, what they're looking for, what would be meaningful. And in many places I work on infectious diseases or things that disproportionately impact people in the global south, so we need to understand what's going on and what would be relevant. So if we think about building an atlas, our lab does a lot of technology development. So I thought I'd give you a little run through of how we've thought about these sorts of things. So some of the technologies I was showing you, like plate-based protocols, get us maybe 1,000 cells. But if we're talking about 37 trillion or the 100 million that John just brought up, it's really hard to imagine how we'd get there. What we really needed were technologies that were going to scale. So to give you an idea of why some of the technologies that we were going to use weren't really going to work very well, I thought I'd do a little analogy right here, because we don't often think about this. So the first time I started doing biology, I thought a 1.7 mil Eppendorf tube was really small. I was taking a pipette. I was using microliters. But if I think about sort of the size of a cell to one of these tubes, and I put a person and I asked this simple analogy that you might get asked on an exam, like how big is a tube relative to a cell compared to us, you'd be shocked, probably to surprise, that it's not a football field, but it's actually 10 football fields on each side, and this can be American football or international football. It's basically the same. So we are putting cells in extremely large volumes. And what this means, if you think about it, is that we're going to need lots and lots of reagents. And so the funny thing, can you click on this? So one of the funny things that I don't often think about, because I come from a non-biological field, is like, what are the sizes of things? And so I really like this video because it helps me to understand how big different molecules are. And so as I think through stuff, I can realize that if I want to make profiling technologies, I have to make them really small and sized to cells. So let's talk about this really quickly. If we think about plates, in version 1, it's 10 microliters, it's got a throughput of about 100. Using some different microfluidic technologies, you can get down to maybe nanoliters, and this gives you an increase of 10, but it really doesn't get you the kind of throughput that you need for the thousands. So in looking at this technology, what we realized was that this capture site right here where we were grabbing the cell was great. It was just everything else around it that was a problem. And so to deal with this, we decided we would get rid of it and try and capture a cell in the smallest container we could, something like a little reverse emulsion droplet. And so this is water and oil. You can think of about it as a salad dressing or whatever you would like, but it's basically an emulsion. And the idea is that because these are 1,000 times smaller, maybe we could get a 1,000-fold increase in throughput. And so if you think about the math now, it works a lot better. If we think about putting cells in little boxes or in little droplets relative to a cell, it's like putting a person in something that's 1.4 meters by 1.4 meters by 1.4 meters. So I'd actually need to squat to get inside of it. So this is the kind of stuff where you can imagine we're not going to waste reagents and we could get to a higher throughput. So how do these technologies work? Well, the first one really was this idea that we would take a tissue, we'd break it apart, we'd capture a cell, but then we'd add a barcoding reagent. The only way you can do things in high throughput is to process everything at once, but tag it early so you can figure out what belongs to whom. And so the idea was that we would add these little beads right here that would carry social security numbers or a unique identifier or whatever you want, a passport number, whatever is your favorite analogy here, as a way of figuring out everything that belonged to a cell, so that even though we pulled everything back together, we could figure out what belonged to each. And I'll say that I'll talk a little bit about DropSeq, and we'll teach some of the methods here, but other technologies have been developed that are similar to this, like InDrop and 10x, which hybridizes some of these technologies. I'm happy to talk about pros and cons of every different approach. I'll just say that everyone is good, everyone is bad. It really depends upon what you're trying to study. So the way in which this works is it really relies upon the idea of a bead and putting a bead with a cell. So these beads basically are containing oligos. These oligos can capture messenger RNA right here. But the most important thing to know about these beads is they have a barcode. So this is like your passport number, your social security number. And these barcodes right here are the same for every single oligo on the bead, but different between any two. So it lets us figure out what information belongs to each. We then develop technologies that come together with these cells. And so this is what the first drop-seq rig looked like, the precursor to the 10X system. And what you can see here is our setup on stage, where we have a bunch of different tubes and syringe pumps pumping into a stage, a bunch of devices that are on a chip. And if, in case you're not used to looking at CADs, this is really the business end right here. So this is where everything comes together. And can you this one? And so what we're doing here is we're bringing together these beads in lysis buffer to capture RNA cells. They're getting isolated, sort of at low density. So sometimes we get a bead, sometimes we get a cell, sometimes we get a bead and a cell. And when the cell and the bead are in there, you can get lysis. The RNA goes around and can get captured. And so at the end of the day, what you get when you sequence is something that looks like this. So we have a bunch of sequencing reads. If we read the 3' end over here, it has this barcode and this UMI. And if we read the 5' end, it tells us about the gene. So what we can do is take this entire mixture of different RNAs, sort by these barcodes, because each barcode corresponds to one cell. We can then go and figure out what gene we're looking at, and then we can use this unique molecular identifier, which is like a barcode for a transcript, as a way of digitally counting things. So at the end of the day, what you end up with is this matrix, which is a number of cells by a number of genes where we're getting counts. And this is what many of you are going to see now when you do single cell analysis. And so what's so great about this is, it lets you do things at throughput. But to figure out whether or not it works, you have to test the technology. And just for a little bit of history, here's how we tested it originally. We took a mixture of mouse and human, so we put the two species together. And what you can then do is you can make a plot like this. And so we're looking at the number of mouse genes here, number of human genes here. If you have a good technology, you'll get a lot of mouse genes or a lot of human genes. If you have a technology that doesn't give you single cell resolution, you're going to get mouse and human because they're going to be lysed together. And if you have a really bad technology that you don't show in public, you get data that's here. Now you only get to see the good data. This is what our technology looked like to start. So we had very low capture, lots of mixing, things that weren't great. By iterating through and doing lots of molecular biology and making things work, we got a little bit better. Eventually we got to a point where we were getting a little bit of contamination, but we were starting to see things that looked pretty good. And through some other iterations, we finally got to a point where we were getting really high quality data. And I'm happy to tell you about all the trials and tribulations here in the late nights. But this put us in a place where we could now look at, get single cells, get thousands of genes, and get a good representation of what was going on. What this enables us to do now is to do the kinds of things that John talked about. To take a tissue like the retina, to collect thousands of genes for thousands of cells. For each one of these little bands of patterns right here where we're seeing genes that are highly expressed that are identifying sort of that cell, that calling card that John was talking about, we can now go isolate them, find markers, and go and look at those cells and characterize them and start to do biology. So if you think about it, it used to take maybe 3 decades in order to find a cell and work your way through it. Now we can do this in a week. But we don't just do this for one cell. We can actually do this for all the different cells at once. And so what we're doing is we're really compressing hundreds of years of research into a week. Now, the problem is that we don't want to do this to figure out what's happening in the retina. We want to do this to figure out what's impacting people's health so that we can figure out how we can make people live longer and better lives. So in talking, for example, with Sarah Fortune, we realized that there was a major issue with tuberculosis, as you probably all are very well aware, where there's 1.6 million deaths every year, leading cause of infectious disease deaths. Maybe COVID took over for 1 year, but TB is now back, which is not a good thing. And a quarter of the world's population is latently infected. But in Boston, as came up before, we don't think about this as much because the disease burden is really disproportionately in South Africa, or Southern Africa and Southeast Asia. If you think about the kinds of technologies you would need to go do work here to figure out what was going on, you would need something that's deployable, scalable, that would work with low inputs, and that would be cheap. And in thinking about that rig that you just saw us work with, what you can tell really quickly is that this kind of system doesn't fit easily into a backpack. It doesn't go all around the world. It doesn't work on lots of different things. So one of the things that we worked on with our partner Chris Love was a way of altering this so that we could get rid of all the peripherals, do things in a very simple capture for less than $10 to build some of these chips so that we could literally deploy this anywhere. What this has enabled us to do, and I'll say there are other well-based techniques, I'm happy to talk about pros and cons here. The way in which ours works is we take this array, we add the same beads that you saw before, The beads fall into our individual wells, we load our cells, we seal them, and we lyse, and this gets us basically, at the end of the day, the same data you saw for DropSeq. In terms of capture, geez, transcripts, it looks identical, but what's really important is this is our form factor. And so if we were here doing science with you, as we have with some people at MIDAL in the past, we would have come with a box full of slides and membranes, maybe some clamps, and we would have actually done some of the work on the ground. What this has enabled us to do is to start to work with partners all around the world to tackle things that are important, like to figure out what's happening in hyperacute HIV infections so we can understand changes in what might drive elite control towards new therapies, to figure out what's different in regions of the gut that are inflamed and uninflamed so that we can figure out how to improve outcomes in IBD, like John was talking about before, and to understand things like what's happening in leptomeningeal disease when we use immunotherapy. And you can see that we're going from something where we have a lot of material down to something half the size of a poppy seed to something where we're not even sure that we have cells. And just to give you a sense of how low we can go, we can take those same nasal swabs that you probably had during COVID way too many times that you don't like, and rather than looking for virus, we can wash them in a little bit of media, load them onto our arrays and sequence to figure out what are the different cells that are in your nose, which ones are infected by virus, and what are differences between people with mild and severe disease. So we haven't just done this ourselves, we've taken it all around the world and partnered with people in 30 plus countries on 6 continents at this point, looking at many different diseases. So I'm happy to help talk about things that you might be interested in, save you some trials and tribulations, and to talk through any of the things that might become important. So just to give you a sense of some of the things that we've been learning along the way, with partners like Al and Henrik down in South Africa, we've been able to identify populations of fibroblasts that associates with severe TB disease that might be targetable. With partners like Sims and Zambia, we've been able to find changes in the gut that associate with environmental enteropathy that might explain why we get poor nutrition and what's driving childhood stunting. And it's really interesting, actually. We're seeing a cell that looks like a stomach cell showing up in the small intestine. And so that could be explaining why you might not be getting good nutrients, because this is really protecting you against infection, but it's not enabling you to absorb the nutrients you need. We can similarly partner with individuals right here at MIDAL, like Jet and Sangeeta's lab back in Boston, to figure out what's happening in malaria and to find new targets that we could go after to actually reduce parasite burden. And the science that we're doing globally doesn't just help us there, it actually even helps us in places like Boston, because now we have simpler technologies that we can use to go partner with people at the Dana-Farber to find differences in cancer cell states so we can identify what are the right treatments for you based upon your particular classification. So I'll say that what we spend a lot of time doing, because we do all this global science, is going around and training people and partnering with them and really trying to make sure that they can use these technologies to the problems that they need. So here are some pictures from some various workshops that we've done in Ghana and in Senegal. But along the way, we learn a lot about how to do this, how to engage with people, how to have conversations, and how to help people. And so we've been trying to share that with the community as well, so that others that are interested in doing this work can begin to do the same. I'll say that on the other hand, we've also thought a lot about how to commercialize technologies, because at the end of the day, you want to produce cheap widgets that can go work for people all around the globe. So what I want to do now is I want to put it all together. So imagine we have these technologies. Imagine we blend up a bunch of tissues, we collect cells, and we throw them onto these devices. Can we envision creating a picture of health? Well, what we can do is we can take 10,000 cells or 100,000 cells and collect 2 trillion data points as a way of thinking about the biology of health. And so, can you click on this? So if you think, what we're going to do here is we're going to place cells randomly, we're going to color them by tissue, and we're going to place them based upon how similar they are to one another. And what we're going to do is we're going to let them sort of evolve over time using some social network-like algorithms to sort cells based upon their similarity. And we'll see if this runs, because it's not on my computer. Is it running on your screen? No. Okay. Well, that's really sad. I have a really cool video to show you at coffee break. What I'll say is, at the end of the day, this thing that would have evolved and it's moved and we would have put everything together will result in something that looks like this, where now we can find lots of different groups of cells and the connections between them. What we can do is we can start to look at the different cells that exist. We can find things like cells that only come from one tissue, like neurons, other things like monocytes that show up in the bone marrow. And in the middle right here, what we find are a lot of immune cells, because those are the things that unify and link together our tissues. With these sorts of atlases, what we can do is we can start to ask questions like, what changes in disease? So the atlas that I just showed you is actually from a non-human primate that was infected with SHIV. where we had many animals that were healthy controls, as well as those that had been infected and then treated with ART. And what you can see is that there are major differences, even when we have suppressed viremia, with different populations emerging. This gives us an idea of why there might be increased inflammation, increased predisposition to specific comorbidities. So, you know, one of the great things about this data, as we pull it together, is that we can share it with the world. And so, you know, we, as well as many of the other people within the ecosystem, started to create these portals that share lots of different information. What we discovered, as John brought up before in the context of COVID, is that this data isn't only useful once. It's useful because as you create more references, you can use that to understand what you're looking at. So when a new virus emerges like SARS-CoV-2, we can take some of the tissue from the atlas that I just showed you, look specifically at the lungs, figure out which cells express the entry receptor ACE2 and TMPRSS2, the accessory protease. And what we find is that really there's a subset of cells within the lung that look like they're likely targets. And so our big takeaway at the end of the day from looking across multiple different tissues that had already been collected early in the pandemic is that likely what we were targeting was secretory goblet cells in the gut, type 2 pneumocytes in the lung, and absorptive enterocyte, goblet cells in the nose and absorptive enterocytes in the gut. We also found that there might be interesting relationships between ACE and interferon, and that there were differences between species that told us that we had to think about which model we were going to use for testing things. Now, we can compare what we learned from doing these early analyses to what happens when we actually go and sequence. And so I told you before, we took nasal swabs and we sequenced individual cells out of them. We built the same sort of atlas that you saw. And if we look and ask ourselves which cells express ACE2 versus which ones had virus, what you can see is that we get this strong correlation right here. So our predictions based upon these references that we had already created actually turned out to be very useful in thinking about the biology of the disease so that we knew where to start right away. Other things that we found that were really interesting were differences in the frequency of different cells in disease and the fact that differences in response associated with differences in severity. So I've talked about the idea of transcriptomics today, but as was brought up in John's talk, over the past 10 years, there have been technology developments that have enabled us to look at everything from the transcriptome to the genome to what parts of DNA are accessible to different RNA or to different features like proteins, and to look at many of them in parallel. We're going to talk about some of these in the workshop. I'm happy to dive into each and the pros and cons. And in parallel, there have been efforts to not just look at these independently, but to understand how they array over space. So there's a ton of technologies that are being developed that will let you look at protein and RNA at different scales and at different resolution. And they're very useful to think about and really interesting to couple together. And we'll cover some of that and some of the discussions we have around our advanced topics. So I'm going to stop there. With 4 seconds left, I'm going to thank everybody who actually does science. Many of them are actually here and are going to be leading the workshop, and I'm very grateful to have the opportunity to work with them. And I'm going to thank our funders and our partners, including all of you. Very excited to be here to learn about your science and to figure out places where we might be able to work together to accomplish more. So with that, I'll take any questions, or we can do that during the break and go to the next talk.
So we're actually a couple minutes ahead of schedule. If anybody has any questions, please feel free to raise your hands for like Alex or any of the speakers. I can bring you the microphone.
Very nice talk. I learned a lot of things today. I'm taking the analogy of these cells with population. How far can you take this analogy? For example, if you can you identify certain individual cells that would be super responders to some treatment of some disease, and then go from there? Are there ways to kind of identify the leaders in the community, so to say?
So it's funny. Actually, one of the studies that we did when I was still a postdoc found that there were these super responders among dendritic cells. It turns out that there are some cells that respond very early and aggressively to bacteria. And I was really interested in this. I wondered whether they were just the smart kids in the front who were raising their hands, the first ones to do it, or if they were really special. And what we found is that actually they really were special. We devised some interesting experiments where we separated them out and we found that only a couple of cells could do this. But what was really fundamentally fascinating to me is that when we looked at how cells responded when they were all together versus when they were separated, without having these early leaders to figure out what needed to happen and to communicate to all of the other cells, the responses that have led to Nobel Prizes on understanding TLR signaling no longer worked. So some of the things that we would have canonically thought would happen didn't happen at all. So what you really needed was you needed a subset of cells to respond, to communicate to others. That led the other cells to have thoughts of their own, which they then communicated back. And so many of the things that we discovered at a population level really depended upon these individual dynamics and communication. And the reason that I actually decided to run a lab was because I was interested, based upon this, in how composition of tissues and communication might actually lead to different outcomes. And so what we find is that in some diseases, there are these sort of super responder cells that look like they would be very interesting. For example, in some of the work that we've done in HIV, we find monocytes that we call super responders because they turn on very strong inflammatory programs and very strong antiviral programs. We also find specific populations of NK cells that seem like they're very important for generating control. So we're discovering them in many contexts. As John brought up as well, what turns out to look like it's the major player in cystic fibrosis is this cell that we didn't know about called the ionocyte. It was something that hadn't been described previously. It was a needle in a haystack. It was found by some of these approaches. And once we found that cell and realized that it was the one that expressed CFTR, which is associated with the disease, it totally changed the way we thought about treating it. So yes, we're discovering things that are rare populations. Sometimes they help us to understand the biology of the disease and what we need to target. And sometimes they give us an idea of what response we want to generate. Now, what we think about a lot that's really cool is that once you know what is the cell that you want, you can now think about things like immunotherapies or drugs that hit specific cellular pathways as a way of increasing that population, or if it's bad, getting rid of it. And so in many places, this discovery helps us to figure out what is the right biology to focus on so that we can get to a place where we can lead better outcomes for people who are suffering from disease.
Thank you very much for all three speakers. Really, I'm very excited to be here and to learn how you were able to separate and isolate individual cells, and then to take a good look at the polymorphism in each individual person or animal. And at the same time, when you have this big, particularly increased number in Asia, among other places, you'll be able to really look at polymorphism at the population level. Now, in response to infection, for example, Ajahn Yongyut was asking about some super responder. But I rather ask about the collective response of different cell types to a situation and the balance of that to influence the outcome of the infection. And this is really ultimately where we could manipulate, where we could shift it one way or the other.
Yes.
How you were talking about contracting 100 years of research into 1 week. So how soon can we manipulate this?
Great.
In a few more weeks?
Great, so we're not asking the easy questions this morning. So I think what you've hammered down is the essential piece of all of it, right? Which is that it's great to break the system apart. It's great to even understand where those pieces are, but what you really need is an integrative understanding of how they work together. Our cells have evolved to have this balance, to have this dialogue, and that becomes really essential. What I can tell you is from the second we start to understand what are the components and what are the signals they generate, it gives us the opportunity to start to make models to explain how they might work together. And I think the advances that are happening with respect to AI and ML and big data are putting us in a place where we could start to imagine models that would tell us about what these individual agents are doing and how they might dynamically evolve over time. I'll tell you that they're models and we have to create better ways of actually systematically testing them. But it's not lost on me that when we think about curing something like cancer, we are not trying to eradicate a particular mutation because that's not what we should go after. What we want to do is we want to clear a tumor. So the big question that we're asking, and I can show you some examples after this, is what is the best way to do that? And it turns out that sometimes it's to act on the immune system to activate T cells because it forces the cancer cells to do one thing, which makes them very susceptible to a drug. And so in many places, what we're learning is that these dependencies can be leveraged. So I showed you the example of pancreatic cancer very quickly. I said different responses at the Farber. But what we find is that one of those response states that we call classical, which is more epithelial-like, responds very strongly to chemotherapy, whereas the basal, which is more stem-like, does not. It turns out if you activate immune responses, you make the tumor very basal and it gets locked in there. But what that does is it makes it so that you create new dependencies on pathways that didn't exist before. You shouldn't use a chemotherapy now, but if you use a MAP kinase inhibitor, you can target that specific state. So by understanding these dependencies and how these cells relate to one another, we can actually exploit that. And so I think that there's this real opportunity to think about how to use the data to look more holistically at how a tissue works, to think about the best points of intervention to sort of get to surgical precision.
Can I have a different kind of question? I would like to congratulate initiators of this field and all who are involved up until now for your openness and for your notion of recruiting more participants and sharing of the benefit. And this is very refreshing in the light of today's intellectual property regulations and pioneers in fields try to maintain some level of secrecy so that eventually the benefit when there is financial return will be kept to small groups. So this, your openness here is really admirable. I would like to congratulate that whole group. But eventually, when new products and new services does come, how do you manage to split the benefit? This is my question.
So it's a great question. So first off, I would say that we aspire to equity, to creating data benefit, to engaging people. We're in a unique position where we are more able to do that because of the privileges that we have. And I recognize that everybody in this room has different privileges, different opportunities, different things that they can and can't do based upon where they are and based upon the people with whom they collaborate when they're generating this sort of data. And so we try and respect that, we try and understand that, and we try and figure out how to help people to accomplish the things they want. I'll say that I'm sure that there's something great that you can do by hoarding data or hoarding technologies, but what I've found in everything that I've done is that I've learned more and have gone faster and have gone further by talking to people, by recognizing the limits of my own intelligence, and by learning from others. I don't know that much about malaria, but I can learn a ton by partnering with people like Jet. I don't know a ton about HIV, but by going down to Durban, I can learn a lot by working with people that, interact with patients, by talking to people that are actually living with HIV. And so that's really what's pushed us forward. And I found that the benefits that come from it accrue to everybody. But unless we're thinking about the people that are actually the ones that are impacted, we're not doing what we need to do. And as my father would say, who's here with me on the trip, you will meet him later tonight, and I apologize for that in advance. My mother's lovely. Just don't talk to my dad for too long. You can't take it with you, so it doesn't matter how much money you have. What's important to me are the people that you interact with, the memories you make, and where you go. And I'm sure it will do well by all of us, but at the end of the day, I think that the reason that we go into science, particularly now when we can go to industry and we can make lots of money and do lots of different things, is that it's because we like interacting with others. We like learning. We like pushing boundaries. And we like thinking about how we can help others. I was going to be a doctor. I wasn't. I ended up being a different kind of doctor, which is not a real doctor, as my parents will tell you. Well, my mother will tell you I'm a doctor. My dad won't. But it's because I wanted this kind of environment. And so I understand why people feel a need for secrecy, but it's really these conversations and finding gaps and finding needs, finding barriers, finding places where we should be tackling problems that really pushes the field forward. It's dumb questions late at night like, why do you think all these cells aren't responding identically? Had over beer that's hiding in somebody's desk on the 6th floor of the Broad that lead us to do new things. It's not just working by ourselves and accepting the status quo. So the way that I think about it is we need to be open, we need to have dialogue, and if we're not talking to everybody and harnessing their expertise, we're never going to get to where we all want to be. Thank you all for coming.
I know. Can I ask one more question? It's just a curiosity.
Don't worry, I'm here all week.
When does diversity in single cell start? I mean, of course, with a single cell organism, you can't have diversity. But when it becomes a planaria or a flatworm, do you have diversity already there? And is it as much as human diversity?
So I would have argued that diversity even starts as a single-cell organism, right? If you think about it, there are lots of different variants in bacteria that lead to antibiotic resistance. We can debate whether a virus is alive or not, but differences in variants that show up in HIV is one of the reasons that it's so intractable as a disease. So there is a ton of diversity, and that's a really important thing for the survival of the species, right? Because it gives it robustness. And, it's really funny to think about where this emerges. We had done some weird stuff with Volvox, which is this organism that can either exist as a single cell or as an assembly of cells. And so there's some really interesting models that you can do by looking at sort of these, let's call them lower life forms, but these more evolutionarily ancient life forms to understand questions like that of when did diversity emerge, how did it evolve. And one of the things that we think about a lot in HCA is humans, but by doing things in multiple different models and multiple different species, we can start to understand how things emerged. When a new cell type showed up, what was its role? How did other cells change in their properties? And why did that give a particular fitness advantage? And that helps us to think about some of the things that we see in humans and how it might work. I think that as we think about diversity, it gets very complicated. As you probably know, you have different mutations in every single cell of your body because it is impossible to perfectly copy 6 billion bases. And so what that means is that every single cell has its own mutations, its own variants. And some of those could predispose you to a cancer. Some of them might lead the cell to live longer and be more fit. And so I think that you could go to this extreme where you could say we will never know all of the diversity. And I think that in a world where we think about things through a personalized lens, like we want to have personalized medicines, that's not going to work. But I think that what we can do is we can find precisions. So we can find groups, categories, broader things that tell us about how cells will respond. What are the really important functional variables? And a lot of the work that we do now focuses not on how do we measure everything, but among all the things that we can measure, which ones are the most important? Because not every single RNA or every single transcription factor is important for determining whether somebody is going to respond to frontline chemotherapy or to checkpoint blockade. And what we have to do now is we have to couple everything that we're doing with models and perturbations and those sorts of things to tell us what's important so that we can use that to guide the interventions that we do. Of course, thank you.
All right, thank you so much, Alex, for your great talk and great comments. So next, I'd like to introduce our next speaker, Associate Professor Varodom Charoensawan from Mahidol University. His lab focuses on computational biology and bioinformatics, single-cell omics, and systems biology. And he will be talking about single-cell genomics in Thailand.
All right, thanks so much. I'm not quite sure how to go from here, after Alex's, excellent talk and fruitful discussion from Professor Thornchai and Professor Yut. Actually, I forgot what I was going to say. Anyway, so I've been tasked to give kind of different kinds of talk. Just give an overview of what we've been doing in Thailand in terms of single cells. So I guess what we try to achieve are twofold, right? First, I think this is just to showcase what we have been doing in terms of single cell, but also in the spirit of human cell atlas in Asia. So we hope that what we have been through, right, could provide some sort of case studies, right, to other countries who might want to doing something similar as well. So I just want to start by giving you a little bit of history of what we have been trying to do. So I think John mentions that HCA started around 2016, right, if I'm not mistaken. So in Thailand, we started to do sort of single cell related in around 2018. So that was when Professor Ponpan and myself got a Newton Fund in collaboration with Professor Suwannalert in Cambridge in the UK. So that was when we realized that it wasn't anyone at all in Thailand doing single cells. So we had to phone call some company just asking them whether they can sell us the machines because no one's selling at that point. And then over the years, so we have gathered a few instruments that allow us to do single cell experiment in Thailand. And this would not be possible without this general funding from both international and also funders in Thailand as well. So just to give a bit of a rewind a few years back. So this is our first run in 2018. So this is the first activities that we've been doing anything related to single cells, and that first run was actually happened here at Faculty of Science, Mahidol University. That I think some of you might be able to see the machines this afternoon. And so the first thing that we did was to not just do it for ourselves. So we organized the first workshop and meetings back in 2018 as well. And that was when we had a big group from Sarah Teichmann in the UK, and Martin Hemberg came over from the UK to have this first meeting here several years ago. In that meetings, we also have just to show you that we have a lineup of different kinds of talks, as well as a 2 days workshop. So back then we were doing 10x and SmartSig 2, which the participant for the experimental track would be would experience this afternoon as well. So in terms of the research questions that we've been engaging, obviously, so I think Ponpan mentioned this as well, that in Thailand, because being in the tropical countries and basically infectious diseases is one of the biggest challenges that we have. So the very first research question that we tackled was on infectious, and this is things to do with human dengue infections. So I believe Varodom, the next speakers will talk about this a little bit later on as well, just to show you some examples that dengue is basically very we see a lot of incidences in Thailand, and one of the key important things of dengue is that you need to know exactly when the fever is going to subside. That's when all sort of hemorrhagic fevers and hemorrhage can happen. So basically, we try to tie up the single cells during a time course and try up with the different kinds of manifestation that you see during the 4 days that we have the data. So this is already published. So and Waradon is going to cover. So I'm gonna not going to go into details. So but the main thing is that we were able to discover specific skin homing markers that we can possibly use to identify when the fever is going to subside. And that's the first publication was done focusing in T cell, and then recently this year we have also published another second papers focused on B cell as well. And this is what Waradon is going to expand in the next talk. Apart from that, we also try to use apply single cell to tackle other diseases, including non-communicable diseases. So in collaboration with our close collaborators, Natini, who is also one of the organizers of this particular workshops. So together we try to use single cell to see whether we can actually observe heterogeneities within the cancer cells. So in this, our pilot studies, we work on the ascites sample of the cancer patients who receive certain treatments. So the key thing is that we observe that within the different kind of cancer cells, we observe different cells with different profiles. And more importantly is that after you give the patients the treatments, some cancers subtypes actually as you expect, right, it's shrinked, whereas there's some cancer types actually expand. So I think it's one of the in our experience, this is one of the first study that show that you can use single cell to actually see different cancer subtypes. In addition to that, just to give you a few examples of the applications. So in collaboration with our colleagues, Ananya, who's also a dentist, she was particularly interested in to use single cell to see different kinds of stem cells that you would see in dental pulse. I think also in collaborations with lots of people here in this workshop as well. So that's basically a few applications that we have used single cell in Thailand. So over the years, I think it has been a few years, since we start doing this, and we keep expanding our network, and basically more and more people, are getting involved in single cell in Thailand. So I try to put together a list of, our collaborators, but also those who that, we realize into single cells, and I try to group the application into, different groups like this. And by the way, this is not an exhaustive list by any means, so I might drop, some names out. So I apologize in advance. So obviously, immune systems and, against infections, we have Ponpan, who's leading the group. But also we have Professor Gorpon, Waradon, Ananya, and so on and so forth who work on this. So another groups, there's people who are interested in, stem cell and organoids. So there are quite a few people who start to, use single cell to, try to study the heterogeneity within, the organoids, which is supposed to be similar to organs, right? And including we have Professor Niti and Pan, who's also in the audience, who work quite a lot on this as well. So you might want to talk to them if you're interested in. And of course, we have lots of people who work on tumor and tumor heterogeneities. And quite recently, we try to tie up long-read sequencing with single cells. So I don't know if my colleagues are here. So Professor Thidatip and Professor Pirun, they're expert in long-read sequencing. So we try to see whether we can use long-read sequencing to actually expand, to see the heterogeneity even further. So I just want to shout out, these people who are also in the audience and being part of this organizing committee as well. All right, and as Alex and also John mentioned that after single cells, right, the next steps is to expand to see the heterogeneity, not just in the population, but actually see in terms of spatial information where exactly it is. So we are slowly trying to initiate spatial transcriptomics in Thailand, and just want to show you one of the examples that we have the technology that we have our hands early access to. So just want to point out that this is, for example, this is the melanoma tissues that are H&E staining. So if you ask pathologists to have a look at this, they will be able to tell you where the cancer cells situated. But that's pretty much what they can do. But if you use some technique similar to single cell, as I mentioned to you, right? But instead of dissociate the cells, but just keep the cell there, right? But then you put the barcodes onto the cell right there instead. You can actually overlay the expressions information with the actual cells in the tissues, and then if you combine them together, you will see that actually in terms of expressions, what you see that they look quite similar in terms of H&E stainings, actually quite different in terms of expressions. So this is in collaboration with Professor Manop, who is one of the key leaders in terms of our influencer in terms of cancers and precision medicine in Thailand, and our teams who spend a whole week in Singapore with I don't know if Sean is here. I just want to keep a quick shout out because he put up with us for the rest of the week for that. So just one slide to show what we try to move into. So this is again the H&E stainings that you can say this is the malignant cells, this is probably benign cells, but when you superpose the expression data, you can actually see that they're quite different. But also more importantly, within the what pathologists see as cancer cells, you see two subtypes of cancer cells. One with kind of typical melanoma cancer cells, but also we see sort of proliferating cell cycles, like melanoma, which is kind of at the interface between the cancers and benigns, right? So I think this is kind of just to showcase that you can see a lot more than H&E with gene expressions. So that's kind of where we're moving to. So just for the rest of my talk, I just want to maybe give a little bit of overview of how Thailand as a country, we got involved with HCA from the beginning. So I think Alex and also John mentioned this already, that HCA is really active, right, in terms of bringing equity to being this access to the technologies to different countries. And I think that was 2019. I got an email from Alex. I wasn't sure whether that's like a hoax or fishing or something. but then he offered to, I think we have a call, right? I mean, have an online call, something like that. And then at the end, I was fortunate to be, in that in person in Ethiopia to have a meeting with Alex and the rest of the team. So this is the photo that I asked from Christine's, all right? So I looked through, I think Alex was there, right? Christine's and Elliot, if I identify you guys correctly. Surprisingly, I couldn't find myself. So I was pretty sure that I was there, but also, well, I tried to understand what happens, but I didn't see Shyam as well. So I think me and Shyam might, just, talking about something else that, I can tell you about later on. So yeah, that's basically, I think our first involvement with HCA from the beginning. That was me giving a talk on something related to the setting up this kind of things in the limited resources and so on. So I just want to, after 4 or 5 years, so I just want to quickly recap what I talked about those days. So I think, being in sort of, I guess we kind of caught in the middle between, developing and developed countries, right? Which is same as many countries in Asia. So there's also a lot of barriers and also opportunities that I think we might be able to learn from here. So obviously, funding is the size of funding is different in different countries, and obviously it's not very easy to set this kind of high tech and quite expensive stuff. So what we have been trying to do is just to group together. We work as a team, and to be honest, we put together a lot of different grants together in just to make this happen. Together with Ponpan and many other people, we trying to bring together, like as well. Basically as much money as possible, to keep this up. Bureaucracy. This is something that we experience a lot, in Asia in particular. When we bought like the first, the first instrument or the next instrument, I got asked like, why would you have to buy something that nobody else in Thailand was doing? Because it's just too much burden for like the people who have to deal with something that they haven't done before. So it was quite difficult back then, but then yeah, that's something that we have to go through. But I would say like in terms of supporting and facilities, I want to say that I'm forever grateful to Mahidol University because at that point when we realized that we need these facilities, right? We got some funding from the university, but also Faculty of Science provide additional funding to make the facility that we have right now happens. And I think that's probably the reason why we got into to be linked with HCA in the first place, because we have this capacity to do the experiments. And I would say that it's very important to have the research communities and public engagement. So I think as a scientist, it's very important for us to relate what we're doing to the community, on what exactly we do and how that's going to affect their life and so on. So Ponpan mentioned that we, in terms of research, we are actively involved in the project called Asian Immune Diversity at Last. So I'm not going to go into details, but that's exactly what we got from the meetings in Ethiopia with Alex and with Shyam, so that we got the funding from Trans-Zuckerberg Initiative for that. And as Ponpan mentioned, that us together with several countries in Asia, we try to put together like the first healthy reference map of population in Asia, covering millions of cells, right, from different populations in Asia. So I just want to end my talk by saying that it's always, when you talk about diversities, right, it's always diversity within diversity. So this is where we are right now, right, Thailand. But so many of you, probably this is might be your first, second time in Thailand. We situated right in the middle of this peninsula, right? We are right now at Mahidol University. But actually, in an even small country like Thailand, we have such a wide varieties. And in the north of Thailand, you can see like people dressed differently, and geographically, and also historically, genetically, we are different. So what we try to do is to understand diversity within diversity by collecting sample, not just from Thai population in Bangkok, but to cover like populations from all over Thailand. But in return, right? We're not just there to collect the sample from them, but we also expanding, disseminating our single cell capacity to different part of Thailand as well. And I think in fact there's a representative from all of these universities in this workshop as well. So that's what we're trying to do as a country. So this is really the end of my talk. I guess Thailand as a country, we're really proud to be, being part of the, like the HCA community from the beginning. Well, I mean, not the beginning of HCA, but quite early. And this is our, what we have 2 years ago, right? This is where we are right now. So I don't have a photo to show you here, right? So I try to use Copilot to try to, give us impressions of what we're going to be in these meetings. So I asked to create an image of participant, right, and instructors, and so on. And you can see what AIs think about in terms of single cell, right? So you can see if cells were to be this big, right, I think it would be much easier to study. But anyway, yeah. So I hope you guys enjoy the rest of the week, and I like to thank all the supporters and sponsors for this. Yeah. Thanks so much. Thank you. Yeah.
All right, thank you so much for your talk. Next, please welcome Dr. Waradon Sungnak from Mahidol University. He is an immunologist specializing in single-cell omics analysis of the human immune response in health and disease, and his talk will be on single-cell genomics in infectious disease research.
Thank you, Vim, for the introduction. So it's my pleasure to be here today, and I'm assigned to talk about the application of single-cell omics research in the context of infectious diseases. And I think I'm going to cover my experience ranging from my years during the postdoc years until now that we are doing research using single-cell omics in the context of infectious disease research, and looking forward to the future. So speaking from the perspective of immunologists, so we think it's quite reasonable that heterogeneity, diversity is something that single-cell omics usually give us information, right? So it's something that we kind of familiar as immunologists, because you can imagine that the immune system has to deal with a wide variety of pathogens all the time, and the pathogens can come in different size and shape, right? Like as small as viruses or as large as worms. So it's reasonable that we have to have the system that can deal with such a wide variety. So we're supposed to have a different immune cell with different effector mechanism, with different recognition processes in such a way that the responsiveness would be the most effective for a particular pathogen. And this is why, even before the age of single-cell omics, we know beforehand that the immune cells are quite diverse. So they're diverse in terms of the development, diverse in terms of the effector function or its regulation. And immunologists are quite obsessed with heterogeneity. We're so obsessed with the subset of the subset of the subset of the T cell, right? Every, new, publication, there would be a description of a new, novel subset of this particular dendritic cells or this particular monocyte that have these very specific spatial function to deal with this specific worm. So the advent of single-cell omics has become something like a dream come true for immunologists, because now we can actually play around with heterogeneity and diversity, and then we can better understand how the immune system actually tailor the response to specific pathogen of interest. And I think this kind of concept of deciphering the cell identity is something that Alex also covered. So we can also think about this as, I think this nice review from Alan Wagner, so a friend of mine who was also with Joseph and with Gap. So we can think about the cellular identity as a sum of vectors, right? When you think about physics. So the cellular identity is actually the sum of different aspects of cellular processes that could be the consequence of things like responses to environmental stimuli, the location, or their stage in cell development. So when we see the profile, when we see like the result that we like to analyze, then we need to decipher like what could be the factors that would eventually sum into this eventual identity. And in terms of decomposing those aspects, we can think about, so typically we first think about discrete cell type, right? But then it can also be a more like continuous phenotype, or the stage in the cell cycle, or the stage in like their developmental trajectory. So these are all the aspects that like we would think about when we see or when we have a single cell omics data, and then like think about like what kind or what direction we would decompose our single cell data set into this aspect that would eventually explain the biology of the data set that we have. And this is something applicable in the context of infectious diseases as well, because in our immune cells there would be responses that eventually when we profile the cells, then we might better understand like the processes here, and we might better understand how the immune cells deal with the pathogen. So in the next couple of minutes that we are going to continue, so I'm going to talk about like two infectious disease systems. So one is SARS-CoV-2 and the other is dengue virus, so something that I've been working on. So the first one that I like to mention is the work that we've done when I was a postdoc in cell attachments lab at Wellcome Sanger Institute in the UK. So this was, I think, around, so the project started around February 2020, so a couple of months after SARS-CoV-2 emerged in China. So at the time, the cases were only in China, maybe some cases in Southeast Asia, but then no cases in Europe yet. So we didn't really have our hands on the infected samples yet. But at that time, Sarah and I had a discussion whether we could leverage the existing human cell atlas, so like both the one that are already publicly available or the data sets that like are not published yet, but we have a network of scientists that we could have some access to the data sets. So we think that what about just surveying this human cell atlas data set and then take a look at expression of viral receptor and genes that are associated with viral entry. So we kind of survey a data set across different tissues and like found some interesting patterns. But I think one of the highlights that we have was that we found the high expression of viral entry genes to be enriched in upper airway, especially in nasoepithelia. So that was something that was not quite established at the time. So at the time, I think the predominant idea was that expression of the receptor was in the lung, potentially conferring the pathology, right, the pneumonia phenotype leading to severe disease and death. But then we found the expression of this receptor to be quite higher in the nasoepithelia and the upper airway. So we think that this could be important for the efficient transmission of the viruses. So this work has been independently discovered by other groups as well, including the work from Alex lab that he just mentioned. And I think now we understand more, particularly like in ciliated cells in the nasoepithelia as a reservoir for the virus, so potentially being important for the point of transmission. And I'm going to loop back in a bit that like what's going on in the nasoepithelia is also, or potentially also important for the outcome of the infection, whether you get infected or not, or whether you get severe disease or not. So before we get to that part, so I'd like to mention another work that I'm also a co-lead in this one. So I think a couple of months later, we actually got our hands on the infected sample. So we initiated the experiment by looking at the systemic responses by checking the blood from the donors with acute infection with different disease severity. So we kind of uncover the immune components that are elevated upon infection, and certain components that are associated with asymptomatic or mild disease versus a component that are associated with more severe or critical diseases. So we kind of uncover certain components like specific subset, monocyte, or like the tendency toward megakaryocyte differentiation and platelet activation. So probably related to thrombosis, like blood clotting found in COVID-19 patients, as well as certain immune components like like T cell phenotype associated with more severe disease, or like IgA 2 positive plasma cells associated with asymptomatic or milder disease. So these are what's going on that we can tease out with respect to systemic responses in acute infection across different disease severity. And then, so I'm going to mention some of other works that I have some minor contribution in, so I'm not the lead author in this one, but just some minor contributions. So later on, we asked whether we could understand why kids tend to get less severe disease. And this time, we look only in, like the blood systemic response, but also in the local response, which is in the nasal epithelia, right? So we mentioned that based on that initial paper, we were kind of interested in the events in the nasal epithelia. So I think the key highlight here is that in kids, we found that even at a steady state, they have type 1 interferon response gene expression even before viral infection, and upon infection is highly elevated. And this is as opposed to adults, where they tend to have very low even after induction in the local immune response, but they tend to have the more systemic type 1 interferon responses. So we think that it's not just about type 1 interferon responses being expressed, but where the responses are expressed. And we think that this could result in like more control locally versus like potential immunopathology of the immune responses. And then, so this one also had a minor contribution in it. So this was just out in Nature last week, online version. So this one was in the COVID challenge model, where there were volunteers who were exposed to the pre-alpha viruses. And in this system, we can control for author-confounding factors, and then take a look at what's going on longitudinally, like in a very precise manner. And we found that, like in certain volunteers, there were abortive or transient infection, while there are others who have sustained successful infection, right? So what we found was that in the patients who have abortive or transient infection, there were quite active, like nasal immune responses there, like class 2 HLA DQA2 expression in antigen-presenting cell, and again, right, local type 1 interferon responses. Again, emphasizing that early event in the nasal epithelia could not only just like being important for viral transmission, but could also dictate the immune outcome. So I like to shift gear toward what we've been doing here at Mahidol University, and I think the system that we spend quite a lot of time on is the context of dengue virus infection. So it's a Flaviviridae family, RNA virus, it's a mosquito-borne. So it has a very high clinical impact, and it's going to get worse with the climate change. But what's very fascinating about dengue virus infection, especially for immunologists like me, is that this infection could have, like potential adverse memory responses. So if you are reinfected with the same serotype, you're likely to get protected. But if you are infected with a heterologous serotype, you have an elevated chance of having detrimental outcome. So there's something about pre-existing immune responses that could lead to, like a more severe disease. So something that is so fascinating, and I think it's a system very suitable to be explored by single-cell omics. And the work that I'd like to focus on, so Varodom mentioned some of the work that we have as a series, right, like from our facility and our research group. But the one I'd like to mention here was from the sample from the dengue-free cohort spearheaded by Ponpan, I think, like 15 years ago. So the precious sample that we had was from asymptomatic donors, where I think 179 household members or people who have symptomatic symptoms were, in a systemic surveillance program. So, the nurse would go to their houses doing RT-PCR, once in a couple of days. So over the 5 years, out of this, 180 household member being surveilled, 8 were acquired, as a symptomatic donor. So we got, 8 as a result. So this very precious sample actually reveals some of the fascinating phenotype that, we kind of discover, including, more antigen presentation and more active CD8 responses in a symptomatic donor versus, in symptomatic donor, we tend to see gene expression related to receptor-mediated endocytosis, type 1 interferon responses systemically, and then IL-10 associated expansion of plasma cells. So this manuscript is under review, so in Nature Immunology right now, so finger crossed, wish us luck. So I think, prospect for the future is that, we are not going to be able to just look at the transcriptome, right? So people mentioned the different layers of the omics, correlation to, human genetics, right? Genetic variabilities and contribution of genetic factors toward the disease outcome, and not even mentioning the interaction of our host with the microbiome in the body. And then there's also the whole universe of, the microbe world, right? There's great microbiologists here, some of us, doing microbial genomics, right? So there's a whole universe here that, in the end, we would look more into host pathogen interaction, doing some correlation cross-sectionally, longitudinally, and then, something related to adaptive immune receptor repertoires, TCR, BCR, antigen-specific receptor responses. So something that we actually look into as well. So, within the context of, human cell atlas, outside human cell atlas Asia, there's also some attempt to steer more into the infectious disease. So I'm giving you an example from the meeting last year. Something as a concept right now, maybe we could have, some more sharing or more standardized framework for sample collection or, pipeline of analysis. So in the future, the integration would be more effective. So something that John mentioned, really, as a context of human cell atlas in the healthy issue itself. So there could be something similar in the context of infectious disease context as well. So I'd like to acknowledge people from, our end, Mahidol University and collaborator at Wellcome Sankar Institute, Sarah and Kirsten, and collaborator in, Arthur University as well as the funders here. And finally, also, I'd like to shamelessly, advertise our graduate program. So if your students or, you have students who might be interested in doing single-cell omics research, so our graduate program has a master's degree and PhD degree. So the scale that we have is quite small now, but we are quite motivated, quite ambitious in tackling, challenging and potentially impactful research questions. So if you're interested, you can talk to us more about our graduate program. So finally, so that would be all. So hope you guys have fun with the workshop. So, get the most out of it and, enjoy the time here. Thank you.